I was able to catch some time with Dr. Ashok Shetty, a professor of molecular and cellular medicine and associate director of the Institute of Regenerative Medicine at Texas A&M University. Known for his work in neuroscience research and cell-based therapies, Dr. Shetty is passionate about his work, and in this interview, you'll get to hear an exclusive insight into his most recent, unpublished findings in the exciting field of neuroscience and regenerative medicine. Hi, Dr. Shetty. So I know that your lab is primarily focused on neuroscience research, and your group has been developing clinical strategies to enhance brain recovery. You have worked with a variety of different cells, including human-induced pluripotent stem cells and bone marrow-derived mesenchymal stromal cells to study the mechanism of action of stem cell-derived extracellular vesicles. So I'm very curious to hear your perspective on what you have observed so far about these cells and whether there are some differences.
Okay, so we work on human mesenchymal stem cells as well as human neural stem cells. So with the extracellular vesicles, we started off with EVs derived from human mesenchymal stem cells. So these are bone marrow derived mesenchymal stem cells.
Right.
Okay. So using ion exchange chromatography, we isolated extracellular vesicles, and then we found that they have very robust anti-inflammatory activity.
Right.
Okay. So we tested them in a model of TBI earlier, and where it improved cognitive function and also reduced neuroinflammation. Okay.
Right.
Especially in the acute phase. Then we tested in another model, which is a status epilepticus model. So you may know about status epilepticus. So we use chemoconvulsant compounds such as canic acid or pilocarpine. In rats, we use canic acid. In the mouse, we use pilocarpine to induce acute seizures.
I see. Got it. Okay.
The animals develop continuous seizure activity.
Right.
So that is called as status epilepticus. They have stage 4, stage 5 seizures for a particular period of time, and that leads to cytokine storm in the hippocampus. So that's the most vulnerable region. So we wanted to see whether these MSC derived EVs can suppress cytokine storm. So we induced acute seizures for 2 hours, and then we gave these EVs intranasally.
Right.
So that resulted in suppression of cytokine storm, and also it reduced neurodegeneration. So typically in that model, animals develop cognitive problems.
I see.
A month later, if you do different behavioral tests, you see cognitive dysfunction. But with EV treatment, we did not see any impairments. So it was neuroprotective. So mainly through anti-inflammatory activity.
Got it.
So that's about the MSCs, and we have a patent on that MSC derived EVs, and that has been licensed to a company called Celtex.
I see. Got it.
So they're going to use that for treating Alzheimer's disease. After doing more basic science studies.
Very exciting. That's awesome.
So in the meantime, since I'm a neuroscientist, I'm also interested in neural stem cell derived EVs.
Right.
So we use iPS cells. Earlier we made GABAergic neurons from iPS cells and tested them in an epilepsy model. We found suppression of seizures because of GABAergic differentiation and integration with the host. Okay. Using the same iPS cells, now we generated neural stem cells and then isolated EVs using similar methods, ion exchange chromatography, followed by size exclusion chromatography. Okay. So we get a type of EVs. They have a very interesting cargo, a lot of microRNAs which have neurogenic properties, anti-inflammatory properties, antioxidant properties, and also quite a few proteins which can influence the disease progression.
Right.
Okay. So that paper is under consideration for publication right now on the characterization of iPSC NSC derived EVs.
Got it.
After revision, it's in review right now. Yeah.
Awesome.
Okay. So we tested those EVs in, again, a TBI model.
Right.
We compared MSC derived EVs with NSC derived EVs.
Mm-hmm.
So it turns out that both have beneficial effects.
Okay.
So they can mediate anti-inflammatory effects and prevent cognitive and mood dysfunction in a model of traumatic brain injury. Yeah. So we have two types of TBI models. One is penetrating TBI, where we do craniotomy and hit the brain directly. That's called controlled cortical impact injury. Okay? And the other model is closed head injury, where you just expose the skull and then hit with the stereotactic device. Okay. So, and that model is a repeated concussion model. And in that model also, we found beneficial effects using EVs. These are our unpublished data, though. Okay.
Very exciting. Wow.
The status epilepticus one is published in PNAS in 2017.
Right.
Yeah.
So are these differences?
Another approach we are taking for brain injury and brain diseases is instead of giving EVs intravenously, we give them intranasally.
Right.
Because by giving them intranasally, within 6 hours, they permeate the entire brain. It is really, really amazing. Yeah. So they get into the the cribriform plate. Through the cribriform plate, they get into the subarachnoid space, and then with the CSF flow, they get into the interstitial space very quickly. Wow. And from the interstitial space, they are picked up by neurons and glia. Mostly picked up by neurons and microglia. Microglia, they can phagocytose these guys. Right. And whereas neurons can take up through exocytosis, endocytosis.
Right. Yeah, I saw that paper you just published.
Yeah, astrocytes, not so much. We have found some EVs getting incorporated into astrocytes, but not a lot of them.
I see.
But neurons like these EVs, they actually take up EVs, which is a good thing. So we can deliver cargo directly into neurons, particularly in diseases such as Alzheimer's disease. So we like the intranasal approach. Yeah, because we can quickly deliver these EVs into different types of cells in the brain.
That's interesting. Yeah. What do you think is the, I guess, the mechanism that's sort of drawing the EVs into the neurons? Do you think there's any particular signaling involved or anything like that?
Yeah, I think in case of microglia, it is a simple phagocytosis because microglia, they look for pathogens or any foreign material which comes in, they phagocytose it. And in case of neurons, it is, I think, they take up these EVs by endocytosis. Based on the previous literature, it involves clathrin, clathrin-mediated endocytosis.
Got it. Okay. Yeah, it's very interesting. I saw that work.
We haven't studied the mechanisms, but based on the literature, it looks like that. It is an endocytosis.
Right. Yeah.
Endocytosis.
No, that's fascinating. So I think based on this study, I think, I guess one conclusion you made was that intranasal is the best approach.
If you're targeting brain diseases.
Yeah, that's really the best approach.
That's the best way to do it. Yeah, some amount might go because of gravity, can get into the esophagus or trachea. Sure. But the majority of them will get into the brain.
Got it. Okay. So a lot of people in the EV space have asked us this question, which is dosing, right? And I'm very curious to hear your thought process and also your approach of what would you do in terms of deciding what would be appropriate dose levels, number of doses, or even dosing frequency?
Yeah. I think so. One has to do a dose response study. So that's what we did in a TBI model. We started with really low doses and then went to very high doses.
Yep.
So in our hands, in the mouse model, it's about 15 billion EVs. Equivalent to one microgram, 1.5 microgram total protein.
Right.
That works really well, even with one-time administration. Yeah. So, but it is, you induce TBI and then give the treatment very quickly. Okay?
Right.
Okay. But in the chronic phase, it may be different. So if you want to give TBI at a delayed time point, the whole thing may be different. You have to do a dose response study all over again.
Right, I see.
In a model of Alzheimer's disease, we have a model of Alzheimer's disease also. We are testing the EVs in that too. So there we are giving several treatments, one treatment every month for 3 months.
Right.
To see that, because the EVs carry a cargo, microRNAs and proteins. So microRNAs probably influence certain signaling pathways, have long-term effects, whereas the proteins, the cargo is released and then they have effect, and then their half-life is limited, right?
Right.
So repeated administration may be required in chronic diseases.
Right. 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 Yeah. No, I think
And
that's
one
always
good thing about intranasal is it is amenable for repeated administration.
right. Yep. You mentioned that on your article. I saw that. Yep.
And it is translatable. Any doctor can do that.
Right. Right.
EVs, yeah,
Right.
EVs are pretty stable. They can be stored at 4 degrees centigrade. So the intranasal administration can
Yes.
be performed in a doctor's office.
Yep. Yep.
Uh,
For sure. Yeah, that's definitely a benefit of utilizing EVs as well compared to just the cellular component. There were a couple of publications I've noticed that also use intranasal approach for COVID-related pneumonia. So do you think that is going to be also just as effective?
Yes, I think so. Yeah, I mean, there's one study published. They tested extracellular vesicles
Yeah.
in COVID patients. I think it was done in New York. Yeah.
Yeah.
So, but they haven't provided details about the composition of these
Mm.
EVs. They just said that these EVs are from,
Right. Right.
yeah.
Yeah.
So
Yeah.
nobody knows about the cargo. Yeah.
Right.
Okay.
Absolutely.
And,
Yes.
the study
That's
is pretty superficial. I mean, they looked at only a few things.
yeah.
But since MSCs are working, there have been many clinical trials with mesenchymal stem cells for
Yes.
COVID-19 patients.
Yep.
The first one was done in China, and
Yep.
then the mesoblast did quite a
Hmm.
few
Yep.
cases, and they reported beneficial effects, but it's not been published yet.
Right. Yeah.
So since MSCs have beneficial effects, I would think that MSC-derived EVs will have very similar effects. And
Right.
using EVs may be much safer.
Yeah, absolutely. I think the outlook is very promising, and
Very
the
promising.
fact that
More studies are needed. Yeah, yeah.
yeah,
There
yeah,
have been very few studies using EVs in ARDS models, in animal models.
very true.
I think more studies are required. Actually,
Yeah,
our group at Texas A&M, we have an Institute for Regenerative Medicine, so we are preparing a grant proposal on that. Actually,
and very exciting. That's great. That's awesome. Yeah,
yeah.
yeah. I saw that you wrote an editorial piece on using the MSCs for combating COVID
Yes, yes.
related pneumonia. So I know since writing that article, a lot of things happen. Right, a lot more companies are coming with approved clinical trials for COVID. What are some of your thoughts, or maybe even concerns that you might have about how fast
Uh,
everything is going?
yeah, I mean, so the results are out only for a few studies.
Yes.
The study from China is published in a peer-reviewed journal. Then after that, I haven't seen many published articles. It's
True.
news mostly,
Yep.
right?
Yep.
So we do not know
Exactly. Yep.
their findings. Yeah,
Yep.
so I'm curious to know their findings. But there are quite a few unanswered issues. For example, what is the best time to intubate?
Right. Yes.
So in the first study, the intervention was done at a very late stage. The
Right.
COVID-19 patients having severe complications like pneumonia,
Yeah.
cytokine storm, or ARDS,
Yeah.
right?
Yeah.
So I'm not sure whether intervening very early would be helpful, because in the early stage of infections, we may need our immune system to work harder,
Right.
to reduce the viral load, right?
Right.
So, but in the later phase, what happens is the immune system is overactivated, so we want
Right.
to suppress it.
That's
I think
right.
MSCs, since they do immune modulation, so they are more effective if it is administered in the later part of the disease rather than in very early part of disease. Yeah.
Yeah.
That's
Yep.
what my opinion. But I would like to know the results. And different
Yeah.
people
For sure. Yeah. I think that's a very good
are
point, and I
doing
don't think.
different interventions. Yeah,
Yeah.
and some
Yeah.
people have got approval for doing um, MSC infusions as a prophylactic treatment.
Yes. That's
Yeah,
right.
yeah,
Yep.
yeah,
Yep.
yeah.
I think that will be very interesting to see the different data as you mentioned. But I haven't really saw any studies on like approach, like the timing, as you said. I think it's absolutely critical when you will be dosing the patient. So that will be very interesting to see as well
Yeah.
for having studies like that. Yeah. So I'm always curious to learn about everybody's past and yours as well. So I would love to hear what sort of led you to this area of research, to neuroscience, and if
Yeah,
somebody has influenced your
yeah.
career.
So I was originally trained as a anatomist, neuroanatomist.
Okay.
So I got a master's degree in human anatomy in a medical
Okay.
school. So then I became a lecturer. So I was teaching neuroanatomy. So I got so fascinated with the neuroanatomy, especially the nervous system, structure as well as function. So then I decided to do a PhD in neuroscience.
Right.
So I went to one of the topmost institute in India is called All India Institute of Medical Sciences.
I'm familiar with them. Yep.
So I got a PhD from there. And then after that, I moved to US as a postdoc, continued neuroscience research, then ended up at Duke University. That's a great place to do
Yes.
research.
Yes.
So I stayed for 19 years at Duke.
Wow.
I started off as a postdoc, and then I became full professor when I left.
That's
so it was a very good exposure. My initial postdoc research at Duke was focused on neural stem cell transplantation in brain injury models. Yeah.
Right.
So that's why I was always interested in regenerative medicine.
Right.
And then once I became faculty member, then the stem cell came up. Right. So then I got involved in stem cells because it's so close to regenerative medicine.
Absolutely.
So neural stem cell has been my focus for a long period of time. And I also worked on neurogenesis because neurogenesis occurs through neural stem cells.
Right.
Right. I've done quite a bit of work in neurogenesis. And then, from neural stem cells, now I moved to stem cell derived extracellular vesicles.
Yeah.
So
that's right.
Much safer now.
Yeah.
Right?
No, the field is evolving really quickly.
Oh, it's evolving very quickly.
Yeah.
It's a hot field now, extracellular vesicles.
Yeah. No, I think it's going to be really exciting what we're going to see. So far, with everything that you have done, I know you've done a lot of work. What are you most proud of? And, what do you think is holding the field back right now?
Yeah, I mean, in terms of cell therapy, I think I have taken cell therapy for epilepsy to the highest level. Yeah. So, for example, initially I started doing studies using fetal cells. Okay, originally, the fetal cells. You take out the fetal hippocampus and transplant into the adult epileptic hippocampus to suppress seizures. Yeah. So then, the whole cell therapy field evolved. So I moved to neural stem cells, and then I moved to iPS cells.
Right.
And iPS cells, we could generate very specialized cells which are required for suppressing seizures in the hippocampus.
Right.
And that was the paper I published last year, 2019, in PNAS. And there it showed that we can transplant medial ganglionic eminence-like cells, which are generated from iPSCs, into the hippocampus, which can suppress seizures by 80%.
Wow.
So that is translatable, So that is something, but it will require more time because that FDA has not approved too many iPSC-derived cells for trials, because of the safety issues. Yeah. So once I think safety issues are resolved, and there will be more clinical trials using iPSC-derived cells for a variety of conditions. So epilepsy is among that. So that's one of my long-standing interests, and I have started from the scratch and went all the way up to iPSC-derived MG cells for epilepsy.
No, that's
that is actually a very good finding. Yeah. So we have done now studies in chronic models also, and we are getting very similar results. We can suppress seizures by 80%.
Wow. Very impressive. That's awesome. Yeah. I think the IPS field is also going to be.
And currently, I'm very excited about the extracellular vesicles.
Yes.
So testing extracellular vesicles from different types of cells, MSCs, neural stem cells. I'm also working on astrocyte-derived EVs and microglia-derived EVs. Particularly, I would like to test them in models of Alzheimer's disease because inflammation is a big issue in Alzheimer's disease.
Absolutely.
So if we can control inflammation, probably we can slow down the progression of Alzheimer's disease. If you can't cure it.
Yeah, no, definitely.
Very promising. So that's one of the areas I'm focusing now. Yeah.
Very nice. Awesome. That's exciting. And what do you, I guess in closing, one last question for you is, how do you see the role of MSCs and I guess EVs now as well evolving in the field of regenerative medicine in the next kind of 5 years?
I think they're very promising, but I think what is required is you need techniques to scale up the production of EVs. And after scale up, we need to make sure that what kind of cargo they carry.
Right.
So that controlling the cargo would be very important, I think. So once you know the specific biomarkers which are involved in slowing down a particular disease, then then the treatment becomes more specific. So right now it is somewhat non-specific.
Yeah.
You take out EVs and put it and hope for the best. Okay, but
Definitely.
thousands of the EVs carry thousands of microRNAs.
Yeah.
Thousands of proteins.
Yeah.
So when you do small RNA sequencing and proteomics, you know that. Yeah. So now the big question is, which microRNAs and which proteins are the most important? Yeah.
Right.
The ability to produce a type of EVs having similar cargo and which is beneficial, I think that is the challenge.
Right. Awesome. Thank you so much, Dr. Shetty. This was very, very insightful. And I'm sure I can't wait to share what we just talked about with everyone. I think that everybody in the field is going to be very excited to hear your insights, your experience. This is very valuable. Thank you so much.
Thank you very much. It's my pleasure.